Good morning, students. Welcome to our new class. Myself Manoj Chotia from Raghunath Vidyalaya, Ratangad. हम पढ़ रहे थे नाइट्रोजन साइकिल. नाइट्रोजन cycles is completed in 4 steps, which we will study earlier. Now, this is the sketch of nitrogen cycle. अगर आपकी बुक में है तो बहुत अच्छी बात है, नहीं है तो आप इसका स्क्रीनशॉट लेके और इसको आप कॉपी कर सकते हैं. मैं साइड में हो जाता हूं. ये नाइट्रोजन साइकिल का स्केच है और इसको आप अच्छे से रिमेंबर कर लेना, डिटेल्स कर लेना. नाइट्रोजन साइकिल जो है उसका सीधा सीधा क्वेश्चन पूछा जा सकता है 2 मार्क्स का कि ड्रा द नाइट्रोजन साइकिल और द स्केच ऑफ नाइट्रोजन साइकिल. सो लेट अस अंडरस्टैंड. First of all, atmospheric nitrogen, as you know, nitrogen is found in maximum amount in our atmosphere, just approximately 78%. Nitrogen is not absorbed by plants directly, but by N2 fixation, means with the help of BGA, blue-green algae, BGA, or by symbiotic or asymbiotic association, nitrogen is converted into its components and then after plant absorb that nitrogen. Atmospheric nitrogen इंडस्ट्रियल नाइट्रोजन फिक्सेशन से किसमें कन्वर्ट हो सकती है? अमोनिया में. अमोनिया इज कन्वर्ट इनटू नाइट्राइट विद नाइट्रिफिकेशन रिएक्शन एंड बाय नाइट्रिफिकेशन इट्स फर्दर. यहां पे भी अपन क्या लिख सकते हैं? नाइट्रिफिकेशन. बाय नाइट्रिफिकेशन दिस नाइट्राइट कन्वर्ट इनटू नाइट्रेट एंड बाय denitrification this nitrate convert into atmospheric nitrogen, and atmospheric nitrogen by lightning in the sky also convert into nitrate, and this nitrate come on the land with the help of rainwater, and some of this nitrate going deep in ground with the water, and some nitrate is absorbed by plants, and plants also observe symbiotic, asymbiotic और ब्लू ग्रीन एल्गी जो भी नाइट्रोजन फिक्सेशन करते हैं उस नाइट्रोजन को भी प्लांट एब्जॉर्ब कर लेते हैं. और प्लांट से यह कहां चली जाती है? एनिमल्स में. और एनिमल और प्लांट दोनों की डेथ होने से क्या होगा? अमोनिफिकेशन की रिएक्शन होगी. किसके द्वारा? फंगी और बैक्टीरिया जो डेड एंड डिके के ऊपर न्यूट्रिशन प्राप्त करते हैं. फंगी और बैक्टीरिया वो क्या करते हैं? इनको वापस अमोनिया में कन्वर्ट कर देते हैं और इस प्रकार से ये साइकिल जो है वो चलती रहती है. सो दिस इज नाइट्रोजन साइकिल. मेक इट्स स्केच. नाउ आवर लास्ट टॉपिक ऑफ दिस चैप्टर इज मैकेनिज्म ऑफ बैक्टीरियल इन्फेक्शन एंड नोडुलेशन. बाय बैक्टीरियल इन्फेक्शन रूट्स कैन गेट a node-like structure called nodulation. As we know, rhizobium is a type of gram-negative bacillus bacteria found in the soil. This is gathered just near the legume plant's root or area of the roots of the plant soon begin to form nodules. The process of formation of root nodule take place in the following steps. First of all, this is the internal structure of root, in which this part of root is called endodermis, and this is root hair, and these are the soil particle, and in the inner side of endodermis these cells are of cortex. So first of all, a specific glycoprotein lectin is released by the legume plants. First of all, a specific glycoprotein lectin is released by a legume plant. This lectin attracts rhizobium towards the root. Second, by the effect of plant hormone. By the effect of plant hormones secreted by roots of plant such as auxin and cytokinin, and the node factor secreted by bacteria. Node factor secreted by bacteria and plant hormone auxin and cytokinin secreted by the roots of plant. Effects make the top of the root hair cold like structure. Means these hormone and node factor of bacteria get improved to root hair by a coiled structure, and by coiling this root hair or the cell wall of the root hair is break down from place to place, and then after bacteria enters into the root hair, and after here we can say bacteria, and after entering the root hair, here bacteria is called bacteroid. Okay, so you have to remember bacteroid. Whenever bacteria enters with mucus substances into broken cell wall of root hair, rhizobium bacteria is called bacteroid. After entering into the root or radicle, the root hair membrane, after curling from an infection, thread which slow enter cells and motivate them to increase the DNA content by multiplication. Means बैक्टीरिया जब इसके अंदर एंटर हो जाता है, एंटर होने के बाद में ये अपना डीएनए को क्या करता है, मल्टीप्लाई करता है. हेंस कॉर्टेक्स सेल्स गेट मल्टीप्लाइड. और यहां से फिर क्या होगा, ये एंडोडर्मिस, और एंडोडर्मिस से फिर कॉर्टेक्स सेल में एंटर होगा, और वहां पर क्या होगा, डिवीजन के द्वारा डीएनए जो है वो मल्टीप्लिकेशन करेगा, और द साइज ऑफ नोड्यूल्स इंक्रीजेस बाय सीक्रेशन ऑफ इंडोल एसिटिक एसिड बाय बैक्टीरिया सेल. We discuss about indole acetic acid IAA in our next chapter. Indole acetic acid is a growth regulator hormone which is secreted by bacteria in the cell of plant, and then it increase its size and it infect the cortex cell and make nodule in full form. After the formation of the nodule, the conduction tissue develops and it establishes a connection with the conductive tissue of the root. This nodule is pink color due to the presence of lag hemoglobin hormone, lag hemoglobin protein. And while these are in white or light in the dead cell. Okay, means the color of nodule is pink or red due to presence of lag hemoglobin protein, and whenever the cell is dead, then the color of nodule is converted into white or yellow. Two types of protein have special significance: symbiotic nitrogen fixation. यहाँ पे दो टाइप्स की प्रोटीन पाई जाती है, कौन कौन सी, लैग हीमोग्लोबिन एंड नोड्यूलिन प्रोटीन. नोड्यूलिन प्रोटीन क्या होती है, देयर मे बी ए सेवरल टाइप्स ऑफ दिस प्रोटीन विच इज रिलेटेड टू द स्ट्रक्चर ऑफ द नोड्यूल्स एंड मेटाबॉलिज्म ऑफ नाइट्रोजन एंड carbohydrate. And second is lag hemoglobin in the active and functional nodule. A red pink color pigment is found. A red pink color pigment is found which is called lag hemoglobin. This pigment absorbed O2 at the time of nitrogen fixation because nitrogen fixing enzyme are not active in the presence of oxygen. So this oxygen is absorbed by lag hemoglobin. Hence, lag hemoglobin absorb O2 to prevent nitrogenase from being inactive, and therefore nitrogen fixation is not possible in the absence of this pigment. Okay, so two types of proteins are present: lag hemoglobin and nodulin protein. Nitrogen fixing by symbiosis is the result of the mutual support and expression of the genes also. You have to remember the node gene of plants, the node gene of plants and the node nip and fix. Node nip and fix gene of host of bacteria and node gene of plant. These genes are help in the formation of nodulation. Okay, this is the mechanism of nodulation or bacterial infection. You have to remember, first of all, roots secrete a glycoprotein lectin. This lectin attract the bacteria, and by the secretion of indole acetic acid, auxin and cytokinin, root hair coiled, and whenever it become coiled, then the cell wall of root hair is damaged. By this damaged portion, bacteria enters into the root hair, and after entering in root hair, bacteria are called bacteroid, and then they multiply by DNA duplication and secretion of indole acetic acid, and then after they perform they enters in the cortex and make a nodule like structure or a thick structure is called nodule, and this nodule is help in nitrogen fixation, in which two types of protein, lag hemoglobin and nodulin protein, are present. Lag hemoglobin protein is help in absorbing the oxygen, because nitrogenase enzyme which is helpful in the nitrogen fixation is not working in the presence of oxygen, so lag hemoglobin absorb that oxygen, so nitrogenase enzyme active and fix the nitrogen into its component, that is called nitrogen fixation. And genes, node gene of a plant and node nip and fix gene of a bacteria are also helpful in nitrogen fixation. So this is our chapter. This chapter is our completed. In next video we will start a new lesson. Thank you.